This conference will now be recorded.
Hello students, in today's class we are going to start a new chapter, Cell, the basic unit of life. Okay, so cell, the unit of life, we are going to learn now. Here, in which order we are moving, that you need to get now. See, we have told about plants, right? First we said about the classification. Then we came to the next portion. First thing, diversity in the living world, that we have learned. Then the biological classification we learned, like how we are classifying the plants, animals, and how we are classifying the organism. So kingdom Monera, Protista, fungi, then all the other classification. Then we came to plant kingdom, right? In plant kingdom also different classification we have learned. Now in the same order we are moving now. So now about plants we have an idea, right? After that, we came to the morphology of flowering plants. That we have learned the morphology about root, stem, leaf, inflorescence, flower, fruit, seed, everything we have learned. Now we are moving to cell, because in next chapter, in botany, next chapter, what you need to learn is the anatomy of flowering plants. So before that, you need to learn the cell structure. Then only you can learn or you can understand the anatomy of flowering plants. So if in this order you are learning, means it will be easy to understand the concepts. Okay, so now we are moving to the cellular level. So the basic unit of life we need to learn. And according to NEET syllabus, in this is the 8th chapter, and this chapter you need to learn cell, how, why we are calling it as a basic unit of life, then structure of prokaryotic and eukaryotic cell, plant cell and animal cell, then cell envelope, cell membrane, cell wall, then about cell organelles, what is the structure of each organ and functions of each organelle, then endomembrane system, endoplasmic reticulum, Golgi bodies, lysosome, vacuole, mitochondria, ribosome, plastids, microbodies, cytoskeleton, then what is cilia, flagella, centrioles, the ultrastructure and function also you need to learn. Then about nucleus you need to learn. So here you're getting so many points in this chapter. Okay, and this, based on this, you can learn the animal or zoology side and botany side. In both sides, this will be useful, these terms and all. Okay, so here cell organelles you need to learn in both sides, right? So first we will learn about the cell organelles here in this chapter, and this chapter is very, very important for NEET exam. Okay, this chapter is having more weightage, so you have you can expect number of questions from this chapter. Clear? Now let's move to the chapter. First, what is a cell? What is this? See, in Latin, cellula means cellula. What is that word meaning? Small compartment. Small compartment. Okay. So this is the smallest unit that is showing all the properties of life, and that's why it is called as the structural and functional unit of all living beings. Okay. Structural and functional unit. See, structurally also it is complete and functionally also it's a complete unit, right? That's why, see, if it is not incomplete means single cell organisms cannot exist, right? Within that single cell, it can function, and all the structures, all the needed required structures are there and it is able to function very well, means that single cell can function independently, means that is the basic unit of life, right? That much is enough for the existence of life. So that's why in multicellular or evolved type of organisms we can see multicellular structure. So those multicellular structures, all the cells are working together to perform a function in them because they are well organized, right? So to do the complete function of a body in a multicellular organism, there is a coordinated function of all these cells, right? So structural and functional unit of life, that is cell. The basic structural and functional unit of life is cell. For all the living beings, that is a basic unit. Okay. Then Robert Hooke. Robert Hooke in 1665. He invented cell, but that was a dead Koch cell, right? And next one, Anton van Leeuwenhoek. Anton van Leeuwenhoek, he described the first living cell. Who invented cell? Robert Hooke. But the living cell was first reported by Leeuwenhoek. Okay. Then later, Robert Brown. Robert Brown. Robert Brown described or he discovered the nucleus. Okay. Robert Brown discovered nucleus. Okay. And the invention of the microscope and the improvement of that electron microscope, that kind of inventions help to reveal the structural details of the cell, right? So the structural details in detail, the detailed study of the cell, that made possible by the invention of electron microscope and all, or the modifications of the earlier type of microscope. Then what are all the properties exhibited by a cell? Properties. Properties exhibited by a cell. Actually, cells, they are complex. Okay. They are complex. Why we are saying like that? And they are highly organized structures. Right? It's very complex. And you can see many organelles inside. Internal components are there. Internal components. Right? So structurally, structurally, there are many organelles. Internal components are there in cells. So that's why so many internal components are there, and they all the components are highly organized and well arranged without disturbing the function of other one, and they are working all of those internal structures or internal components are working in a well organized manner, and that's why it is showing a very complex structure, right? And next, all the cells have a genetic blueprint, right? They have a genetic blueprint, right? Or we can say that the cells, they have a genetic constitution in each one, right? So they have their own genetic content, and where it is present, we can say that generally in the case of eukaryotic cell, we can say that it's in the nucleus, right? So the cells, how new cells are getting formed? New cells that will form from the pre-existing cells. So new cells are getting formed from the pre-existing cells. And we can say that cell division. Cell division, that is so here what and all we said, cell is a small component, and that is a structural and functional unit of life. So Robert Hooke in 1665 he discovered cell, which is that was a dead cell, dead cork cell. And Anton van Leeuwenhoek later he discovered a living cell. Okay, and Robert Brown he discovered nucleus. Okay, and what are all the properties we said for the cell? The cell properties we can list out as they are very complex structures and highly organized, because so many internal components are present in the cell, and all these internal components or internal organelles, they are highly organized and well arranged without complicating the function or without creating any complication in the function of the other organelles. And overall, altogether, all the organelles are performing well to make it a functional unit. Okay, to make the cell in a functional way. Then the internal components are present, we said, and the genetic blueprint cell that will be having its own genetic blueprint, or the genetic material will be present inside the cell, and new cells are getting formed from the pre-existing cells, and that's what we are calling as cell division, right? And the cells, they'll get energy or they'll acquire energy, and they'll utilize the energy to perform chemical and mechanical activities. They'll acquire energy and they'll utilize energy, right? They're acquiring energy, and or they're making energy, and they're utilizing that energy to perform all the reactions or all the activities of a cell, right? Then next we need to tell about cell theory. Cell theory. See, as I said, cell is the basic unit of life. Here, this chapter is the basic for all the chapters. If you are understanding this chapter well, about cell, if you are clear in zoology chapters and botany chapters, both sides, this will be useful. Okay, so cell theory. Now we are going to discuss here cell theory. M.J. Schleiden and Schwann. M.J. Schleiden and Theodor Schwann. Theodor Schwann. They proposed this, or they formulated this cell theory. They formulated the cell theory in 1839. Whom? M.J. Schleiden and Theodor Schwann. They proposed or they formulated the cell theory in 1839, and later this cell theory was modified by Rudolf Virchow. Who modified cell theory? Modified by Rudolf Virchow. Rudolf Virchow, that was in 1855. Okay, so we need to learn first who proposed it and who modified it. So Rudolf Virchow modified the cell theory to explain the formation of new cells. Up to that point was not included, and the cell theory we can explain as the first point in cell theory: all the living organisms are made up of one or more cells. All living organisms, they are made up of cells, right? And cell, that is a basic structural and functional unit of life. Basic unit of life. That was the second point. And later, Rudolf Virchow made some modifications that was added as a third point. All the cells are arising from the pre-existing cells. Cells from pre-existing cells. What do you mean by this rising of cells from the pre-existing cells? See, this is one cell. Okay, and how new cells are forming? This is a pre-existing cell, or the cell which is present now. From this, after attaining maturity, what the cell will do? Cell will undergo division and it will form two new cells, right? So from the pre-existing cells, new cells are getting formed. This is a cell division. Okay, so all living organisms are made up of one or more cells, and cell, that is the basic functional and structural unit of life, and all the cells arise from the pre-existing cells. This is a cell theory. Clear? Next, what you need to learn is types of cells. Types of cells. So here, depending upon the nature of nucleus and the basic structure, based on nucleus and basic structure, cells are divided into two types. First one is prokaryotic cell, and second one is eukaryotic. Okay, karyon means nucleus, prokaryotic cell and eukaryotic cell. Okay, so cell that does not have a nuclear membrane. Okay, so there is no nuclear membrane in which one? That is called as prokaryotic cell that does not have a nuclear membrane and nucleoid. And there is no well-defined nucleus. No well-defined nucleus. Okay, so here most of the prokaryotic cell, specifically we can say the bacterial cell, that have a cell envelope, and inside that, like there will be an outer glycocalyx, and there is a peptidoglycan cell wall and plasma membrane, and inside that liquid structure will be there. So the prokaryotic cell, we can represent prokaryotic cell by bacterial cell. Okay, so bacteria cell, blue-green algae, mycoplasma, PPLO, or PPLO means pleuropneumonia like organism. So all these are generally very small cells, and they will multiply. All these cells can multiply very rapidly compared to the eukaryotic cell. Okay, so here you can say all these cells, they vary in shape, size, and all those, right? And the organization, what and all we can say about the organization of a prokaryotic cell. See, prokaryotic cell, we said now different types, like blue green algae, we said bacteria, mycoplasma. So they are showing, like prokaryotes are exhibiting wide variety of shapes, and they are doing variety of functions, right? Wide variety of shapes, and they are performing variety of functions. And we said prokaryotes have a cell wall which is surrounding the cell membrane. In mycoplasma, it is not there. Okay, so exceptions are there in this also. Except in mycoplasma, prokaryotes have a cell wall which is surrounding the cell membrane. Okay, and inside the fluid matrix which is present, that is the cytoplasm. And there is no well defined nucleus in which one, in prokaryotic cell. And genetic material is present. Okay, genetic material is present usually in the normal cell. In the eukaryotic cell, where the genetic material will be present, it will be present inside the nucleus. So it is not present in nucleus. In which one? In prokaryotic cells. So we can say it's loosely somewhere present. So the genetic material is basically naked, and it's not enveloped by a nuclear membrane. It's not protected inside the nuclear membrane. In which one? In prokaryotes. Okay, and we said which one is present here, or which type of cells are there, like bacterial cells and all, example for the prokaryotic cell. So in addition to that genetic material or the genomic DNA, or that single, the genetic material or the genomic DNA, what and all will be there? Many bacterial cells have a circular DNA, very small circular DNA outside or other than the genomic DNA. So these smaller DNA, that is called as plasmids. Other DNA, small circular DNA, which is called as plasmid. Okay, and these plasmid, they'll be having some unique phenotypic characters, like they'll be having some unique features which is giving to that bacteria. Okay, so one of such character we can say, that is antibiotic resistance. Clear? So that this feature is utilized for monitoring the bacterial transformation with the foreign DNA. Okay, so this kind of special features of that plasmid DNA is getting utilized for the monitoring the bacterial transformation. And Here in the eukaryotes, what and all we can say, eukaryotic cell, they have a cell with advanced nucleus. Cell with advanced nucleus. And inside the nucleus, see, there is a nuclear membrane. Nuclear membrane is present. And inside the nuclear membrane, you can see what and all you can see inside the nucleus. There is a genetic material, right? Genetic material is present. Then internal organization of this eukaryotic cell, they are well advanced or elaborate compared to the prokaryotic cell, right? So all the cell materials or cell organelles, all those are present in the eukaryotic cell. Clear? So here you can see the difference between prokaryotic. Here from this you can make out the difference also. You can write the difference of prokaryotic and eukaryotic cell. Okay. And here we are mentioning what? We are mentioning the bacterial cell or bacterial cell structure and the plasmid DNA. All those points are coming here. Plasmid DNA and the antibiotic resistance of that plasmid, and all the features where we are utilizing for the bacterial transformation techniques and all. From for that we are utilizing that special feature of that plasmid. Clear? And from that you can make out the difference of eukaryotic and prokaryotic cell about the genetic material or the nucleus, then cell organelles, and with advanced nucleus, everything that and all the differences between the prokaryotic and eukaryotic cell. Clear? Now, types of eukaryotic cells. See, we said the eukaryotic cells are well advanced, right? So the eukaryotic cells, they are of different types, like two types mainly we can say, plant cell and animal cells, right? Plant cells and animal cells. So here, most of the organelles and all other structures, they are common to both the animal cell and plant cells, right? So the difference between plant and animal cells that we can make out, the cell wall. When we are saying about the cell wall, we can tell the difference. Cell wall in animal cell wall is absent, right? See, animal cells, we know that our body is so flexible, right? The skin, just you touch the skin, it is very soft and it is flexible. We can move. It's not like a uh, something like a plant body, right? It's very soft and it's flexible. So in such bodies, if some rigid material is there, means it's impossible, right? So what we can say, cell wall, that is absent in plant cell. Sorry, in see, this is plant cell. So under plant cell, what you need to write? In animal cell, absent. Cell wall is absent. In plant cell wall is present. Right? In plant cell wall is made up of what? Cellulose. It is made up of cellulose. A cellulosic cell wall is present in plants. And what about the shape of plant cell? In plant cell, you can see a fixed shape, right? Why? Because cellulose, that is a rigid material which is where it is made up of, the cell wall is made up of cellulose, right? So that will give a specific shape, right? But here there is no cell wall. In which one? In animal cell. So there is shape, we can say round or irregular shape, we can say, because there is no stiff material around that. Then what about centrioles? We'll learn about all these organelles. So in animal cells, centrioles are present. This is shape and this is centriole. Centrioles are present in animal cells, but in lower plant forms, in lower plants, we can see centrioles. Centrioles are seen in lower plant forms. Then what about vacuole? In animal cell, you can say like very small. If it is present, it will be very small. Smaller vacuoles you can see. Vacuoles. Okay, very small. But in plant cell, you can see a larger vacuole that may be central, which is occupying around 90% of cell volume. Big central, which occupies the 90% of cell volume. Okay, so these are the differences which you can mention between the plant cell and animal cell. And we know the difference between the prokaryotic and eukaryotic cell. And so that prokaryotic cell and eukaryotic cell, there are a lot of differences. So that differences after learning about all the organelles, cell organelles, we can learn. Okay, so while learning about each and every cell organelle, we'll mention what is present in prokaryotic cell, what is absent in eukaryotic cell, like that, so that after completing the whole points, you can only write the complete differences between the prokaryotic and eukaryotic cell. Okay, there are a lot of differences in the cell wall, in flagella, in cell organelles like plasmid, in ribosome, DNA. In nuclear organization, differences are there in cell size and cell envelope. Everywhere you can see slight changes. Okay, so let's move to the cell organelles now. First one, like now we are moving to the components of a cell. Okay, so components of a cell, that we can categorize into outer covering, like the cell wall and the cell membrane. Which are all the outer covering? Cell wall and the cell membrane. These are the outer coverings of a cell. Then about the protoplasm. Next is protoplasm. Protoplasm, which is the fluid content of the cell. Right? Then inside that, cell organelles you can see. Okay. That means these cell organelles will be floating within the protoplasm, like mitochondria, chloroplast, ribosome. All these things, all these cell organelles will be floating in the protoplasm. Okay, so first we'll see one by one. What is that? First one is cell wall. Cell wall, which is coming under the outer covering, right? So this cell wall, that is the outermost layer. Outermost layer, and this is very rigid structure, right? And it's a protective structure. And cell wall is non-living. Okay, a non-living structure. Okay, and in prokaryotic cell, like bacteria, cyanobacteria, then in eukaryotic cell like algae, fungi, plants, animal cells, and all these uh, cell wall See, we are saying about cell wall. Cell wall, in which one it is present? In bacteria. Then cyanobacteria. Cyanobacteria. In all cells, cell wall won't be present, right? We said the prokaryotic and eukaryotic cell. So here the bacteria, cyanobacteria, then all these are the prokaryotes, right? Then in eukaryotes, like algae, fungi, then plants, which are coming under eukaryotes, right? So all these, they have a cell wall outside the cell membrane. Okay, these possess cell wall outside cell membrane. Okay, so the outer covering, we are saying cell wall and cell membrane. So outer to the cell membrane will be there in all the cells. Outer to that cell membrane, there will be extra structure cell wall, that is for bacteria, cyanobacteria, algae, fungi, plants. Bacteria and cyanobacteria, they are the prokaryotes, and in those cell wall is present. And in algae, fungi, and plants, you know, they have the cell wall, right, which is made up of cellulose. So here algae, fungi, plants, which are coming under eukaryotes, they also have a cell wall. Okay, outer to cell membrane, they have a cell wall. And this cell wall, we can see that is a rigid, cell wall is a rigid structure, we said. And this is giving a shape and size to that cell. Okay, this gives shape and size to that cell, and apart from all these things, it is providing mechanical support to that cell. So cell wall, that is rigid and that is maintaining the shape and size of the cell, and apart from all these things, that is providing mechanical support also. And in plants, we can say the cell wall is made up of 4 layers. In plants, cell wall is made up of 4 layers in plants. So which are the 4 layers? Middle lamella, primary cell wall, then the secondary cell wall, tertiary cell wall. Which are the 4 layers? Primary cell wall, then first we need to say middle lamella, primary cell wall, middle lamella, primary cell wall, secondary cell wall, and tertiary cell wall. Okay, so the middle lamella, that is the outermost layer of cell wall. Okay, middle lamella. Middle lamella. So this middle lamella, that is separating the walls of two adjacent cells. Separates walls of two adjacent cells. So that is the middle lamella between the adjacent cells, or that is separating two adjacent cells. It is present in the middle of two cells. Okay, try to keep it like that. Middle lamella. That is the lamella or the separating wall which is present in between, in the middle of two cells. So middle lamella, that is the outermost layer of cell wall, and that is separating the walls of two adjacent cells. And this middle lamella is made up of calcium and magnesium pectate. Calcium pectate and magnesium pectate. Okay, so middle lamella is separating, or that is separating the adjacent walls of two cells, and it is made up of, middle lamella is made up of calcium pectate and magnesium pectate. Clear? Then next comes the primary cell wall. Primary cell wall, and this primary cell wall, we know that primary cell wall, that is as the word means, primary or the initial wall that develops on both sides of middle lamella. See, middle lamella is separating two adjacent cells. So on both the sides of the middle lamella, this is the first or the initial wall. Initial wall which is developing on both sides of middle lamella, and this primary cell wall which is made up of cellulose. Primary cell wall is made up of cellulose. Okay. Then other things which are present: cellulose, hemicellulose, then lignin and pectin. Okay. So primary cell wall, that is the initial cell wall layer, initial layer which is forming next to the or both the sides of the middle lamella, and this primary cell wall is made up of cellulose, hemicellulose, lignin and pectin. Okay, and this hemicellulose, here we said hemicellulose, right? So what is that hemicellulose? Is made up of pentose. Okay, hemicellulose is made up of pentose sugar. Okay, that, what do you mean by that? Xylose, then hexose, then mannose, galactose, all these. Okay. Then we are moving to the primary cell wall over. Next one is the secondary cell wall. So this primary cell wall is followed by the secondary cell wall. Secondary cell wall. Next to primary cell wall, what is there? After that, secondary cell wall. And secondary cell wall is made up of cellulose, hemicellulose and lignin. Cellulose, hemicellulose and lignin. Okay. Which one? Secondary cell wall. Then what about the tertiary cell wall? Tertiary cell wall. This tertiary cell wall, that is present beneath the secondary cell wall. In which one? Secondary cell wall in tracheids. In tracheids. Okay. Then now we say cell wall is composed of 4 layers. In which one? In plants, middle lamella, primary cell wall, secondary cell wall and tertiary cell wall. Each one is made up of what? That also we have learned. So next we need to learn about the functions of cell wall. Functions of cell wall. So what are all the functions of cell wall? See, the cell wall that reduces the transpiration rate. Transpiration rate. Cell wall reduces the transpiration rate, and how it is happening? That is due to the deposition of cuticle. Cuticle deposition is there, which is making impermeable to water. Water will become impermeable, right? So it is reducing the transpiration rate. Okay, then on cell wall some pits are present, so that is helping for the intercellular movements. Okay, that is helping for the intercellular movement of materials and cytoplasmic continuity. That is maintained with the help of these pits which are present on the cell wall. Clear? And cell wall is providing shape and mechanical strength, that we have already told. Shape, it is providing shape, then mechanical strength, mechanical support to that cell, and that is protecting the cells from the chemical actions. That is a protective layer, right? It is protective layer and it is protecting the cell from the chemical action. So that's about the cell wall. Next we need to, so what do you need to learn? The structure, like how it is made, and if it is having any layers, or it is made up of, it is composed of what, and what is the function of cell wall. These are the things you need to learn about cell wall. Next is about cell membrane or plasma membrane. Or plasma membrane. Cell membrane or plasma membrane. Here, the cell membrane or plasma membrane, that is also coming in the outer covering, right? It is the outer covering in the cells which doesn't have a cell wall. Okay, so here that's a very thin structure, and it's a very delicate structure, and it is having elastic properties. So it is having elastic, and that is the living boundary, right? And who gave this term cell membrane? This term cell membrane, that was given by Nageli and Kramer. C. Nageli and C. Kramer. C. Nageli and C. Kramer, they gave the term cell membrane. So cell membrane, that term was given by C. Nageli and C. Kramer. Okay, and different models of the cell membrane was proposed. It was proposed, which model? Bilayer model. Bilayer model. Different models were proposed. Okay, so in this bilayer model of cell membrane, that was proposed by Daniel and Davison in 1935. In 1935, who proposed the bilayer model of cell membrane? Daniel and Davison. They proposed the lipid bilayer model of cell membrane. See, as per their model, as per Daniel and Davison model, plasma membrane is made up of 3 layers. Okay, so according to this Daniel and Davison model, there are 3 layers for this plasma membrane, and that is a lipid layer that is sandwiched between 2 layers of protein. A lipid layer between 2 protein layers. Okay, so a lipid sandwich layer. Lipid is sandwiched between 2 layers of protein, according to Daniel and Davison. Okay. Then in 1959, Robertson proposed unit membrane concept. Robertson proposed unit membrane concept. Unit membrane concept. Who proposed? Robertson proposed unit membrane concept based upon the electromicroscopic study on myelin. So electron microscopic study on myelin. Based on that, Robertson in 1959 proposed unit membrane concept. Okay, so as per Robertson's model, all the biological membranes have a unit membrane construction. According to Robertson's model, all the biological membranes have a unit membrane construction. Okay, then in 1970, new invention. See, first from 35, who Daniel and Dawson, they proposed lipid bilayer model. Okay, then in 1959, Robertson, he brought the unit membrane concept. Next, in 1970, Nicholson, Singer and Nicholson, S.J. Singer and G.L. Nicholson, they proposed which one? Fluid mosaic model. Fluid mosaic model. Okay, so as per the fluid mosaic model, plasma membrane is composed of phospholipids. According to this, plasma membrane is composed of phospholipids. Okay, then phospholipids, then extrinsic proteins, that means peripheral proteins and integral proteins. That means extrinsic and intrinsic proteins are there. Then Singer and Nicholson fluid mosaic model, that is different from Robertson's model in the arrangement of protein. See, Robertson, he proposed unit membrane concept, but in 1917, S.J. Singer and G.L. Nicholson, they proposed a fluid mosaic model. According to their model, plasma membrane that consists of phospholipids and proteins, that means extrinsic proteins and intrinsic proteins. Okay, intrinsic protein means integral protein and extrinsic protein means peripheral proteins. So all these are present, and the arrangement of these proteins, they are different in this model, not like as the Robertson's model explains, that this fluid mosaic model arrangement, that is different from Robertson's model. In the case, compared to, when we are comparing both the models, the arrangement of protein, that is different in both. Okay, and as per fluid mosaic model, correct sequence of plasma lemma. Okay, plasma lemma sequence. In this, in fluid mosaic model, the correct sequence of plasma lemma is protein, lipid, protein and lipid, then lipid. So the fluid mosaic model, that is the most reasonable explanation, or the most reasonable explanation of the structure of plasma membrane. So this explains the structure of plasma membrane properly. Okay, so these are the models proposed, and this you need to learn. Next, the function of cell membrane. Functions of cell membrane. We need to see that also, functions of cell membrane. See, cell membrane that is performing several important functions like exocytosis, endocytosis, phagocytosis, passive transport, active transport. So all these exocytosis, exocytosis. Let's see what is this exocytosis. See, that is the ejection or the secretion of substances from the cell. Exocytosis. It is ejection from the cell. Ejection from the cell. So how it is taking place? A substance which the cell want to eject, that will be covered or that will be enclosed in a membrane or membranous vesicle, and that fuses with the plasma membrane. See, here is a plasma membrane. A substance want to get ejected from, or a waste substance, something that will be covered in a membranous vesicle, and then it will come and fuse here. Okay, here it will get fused with the membrane, with the plasma membrane. Then this plasma membrane ruptures. Okay, here it will rupture, the plasma membrane ruptures, and that releases the substance outside or the exterior side. Okay, next, endocytosis. Endocytosis, or we can say pinocytosis. This is taking place when the plasma membrane sink beneath an external fluid drop. Okay, so the membrane edge that fuse and that forming a fluid filled vesicle. Okay, a fluid filled vesicle is forming, and then that's how it is taking inside the cell. Next is phagocytosis. Cell eating. Okay, cell eating, we'll call it a cell eating. So this is taking place when a large external particle is surrounded and that becomes enclosed in a vesicle. Okay, so this is cell eating, we can call large particle. Okay, then the passive transport. All these are the functions of cell membrane. Passive transport. Passive transport means there is no direct involvement. In passively, it is participating in the transport. So that is taking place when a substance moves across the membrane from the higher to lower concentration level. Here no energy is required. Passive transport, no energy requirement. And here substance moves across the membrane. Across the membrane, substance is moving. So across the membrane, the substance is moving. That means how it is moving? From a region of higher concentration, it is moving to a region of lower concentration. Then what is active transport? Another function is active transport. Means that cell membrane is actively participating in that movement. Okay, so substance is moving against the concentration gradient. It's not from the higher concentration to lower concentration. Wherever the substance is required, so like that it is moving. That means it may be against the concentration gradient. Okay, so that time against the concentration, it is moving, means there is an energy requirement. Okay, so against the concentration gradient, and here energy requirement ATP is utilized for that energy requirement. Clear? So these are all the functions of cell membrane: exocytosis, endocytosis, phagocytosis, passive transport, active transport. For all these, membrane, cell membrane is helping. Clear? So now we have learned like what is a cell theory, then types of cells like prokaryotic and eukaryotic cells, then different types of eukaryotic cells, then the component of cells. In that we have learned the cell wall and the cell membrane, right? The outer covering we have learned. And about the protoplasm and the cell organelles we'll be learning in the next class, and after that we'll go for the previous year questions also. Okay, so almost the previous year questions here, whenever I'm explaining the topics, all the concepts or the previous year questions I'm explaining here, I'm not mentioning for this which year all the questions came, but here about the passive transport, energy required or not, and how it is working, and the components of cell wall, middle lamella, primary cell wall, everything I'm explaining, all these are previous year questions. Okay, so again specifically we'll go to all the important questions which they have asked in the previous years after completing all the topics. Okay, thank you.